would like to welcome Dr. Shalini ma'am, who is head of department of our biotechnology in our college, for the welcome address of the esteemed guest. Over to you, ma'am.
Thank you, Dr. Rohit sir. Namaskar. On behalf of Dolphin PG Institute of Biomedical and Natural Sciences, I extend a warm welcome to all present today to join this distinguished lecture on computational insights into biological networks by Dr. Karthik Raman. I extend warm welcome to you, sir. Thank you for joining us. I take this privilege to give a brief introduction about the Institute. Started in 2002 with 4 programs, today Dolphin PG Institute of Biomedical and Natural Sciences offers 22 job-oriented undergraduate and postgraduate programs in the field of paramedical, life sciences, biomedical sciences, education, and commerce. The institute is affiliated by NAC with B++ grade in second cycle and is also recognized by UGC under Section 2F. Institute has a team of qualified and experienced faculty members appointed as per UGC norms, supported by a dedicated team of administrative staff for smooth day-to-day functioning. I am happy to share this that till date, 9 students have been awarded gold medal by the affiliating university. Besides conferences, workshops, hands-on trainings, extension activities are also a regular feature for all-round development of the students. This lecture shall be a learning experience for all the participants as they will be getting an opportunity to listen to this distinguished speaker. I once again welcome Dr. Karthik Raman and wish this seminar to be a great success. Thank you, sir, for joining us. I now welcome Dr. Ashish Raturi for introducing our speaker of the day. Sir, please.
Thank you, ma'am. Am I audible? Okay. Very good afternoon to all. And I'm very much delighted to introduce today's speaker, Dr. Karthik Raman. And in the beginning, I would like to thank Dr. Raman for accepting my invitation for this distinguished lecture. Dear friends, Karthik Raman is an associate professor at the Department of Biotechnology, Bhupat and Jyoti Mehta School of Biosciences, IIT Madras. Karthik's research group works on the development of algorithms and computational tools to understand, predict, and manipulate complex biological networks, broadly spanning computational aspects of synthetic and system biology. Key areas of research in his group encompass microbiome analysis in silico, metabolic engineering, biological network design, and biological data analysis. Karthik also coordinates the Center for Integrative Biology and the System Medicine at IIT Madras and is a core member of Robert Bush Center for the Data Science and Artificial Intelligence. Dr. Karthik teaches courses on computational biology and system biology at IIT Madras and has also authored a textbook on computational biology. Dear friends, really he is the cosmos personality of the intellectual domain of this country and the man who has a quest to reveal the unexplored wonder of biology through his computational skills. So, without any further ado, I would like to invite Dr. Karthik Raman to deliver his talk on computational insights into biological networks. Yes, sir. Please, mic is over to you.
Hi, good afternoon, everyone. I hope I'm clearly audible.
Yes, sir.
Excellent.
Yeah.
So I hope, can you make sure that my screen is visible to everyone? The
Yes,
slide
sir.
deck?
Is it really good? Yeah, it's perfect.
Okay, let me go on. So thanks everyone. Thanks a lot, Dr. Ashish, for this kind invitation and introduction. Very happy to be talking about this. As one of you mentioned in the beginning, it's computation and mathematics have become a very integral part of biology, and I'll also be emphasizing the same. Please do stop me at any point if you want to have some additional questions or discussion. I would like it to be a little more interactive rather than just a monologue. So let me get going. So what is the use of computing in biology? And computing and biology have had an exciting history, and this is a great time to work or study at the interface of both. And there are many biological problems that cannot be solved without a computer, like genome sequencing and so on. And protein folding still remains unsolved, even though you have AlphaFold making big inroads into the problem. And modern science has become increasingly computation or simulation driven, and your in silico simulations can predict the outcome of real life experiments fairly well. And biological data analysis today is a major challenge that is faced by both biologists and data scientists who work in the area. But we live in exciting times, and almost a decade ago, the Nobel Prize in Chemistry was awarded for the development of models. Right? It was not for finding a new reaction or for a new catalyst or anything like that, but for basically building computational models. And the citation included this wonderful statement saying that today the computer is just as important a tool for chemists as the test tube. And I would just score out chemists there and put biologists, simply because I think it's very important for practically every field that's out there today. And to add to the quote that the first speaker said today, so all biology is computational biology today. And this is a very provocative piece written by Florian Markowitz. And he says that biology is turning into a quantitative science. How many of us would have thought of that, right? Because we typically have to choose between biology and math in like class 11 or something like that. But today you see that biology is so important for mathematics is so important for biology and so on. Okay. And there are various flavors of quantitative biology. You did hear about bioinformatics being mentioned, right? So that is sort of, I would say, is the intersection of IT with biology, wherein we focus on databases, on software tools, and so on. And then there's computational biology, which is sort of intersection of computer science or algorithmics with biology. Revolves around algorithms for solving biological problems. And then you have systems biology, which is basically where I work in, which involves the application of engineering, mathematics, or physics concepts and principles to biology. And revolves a lot around modeling biological systems and networks. So, quick overview of our lab. So, we are an interdisciplinary group focused on computational approaches to understand and manipulate biological networks. We focus on scalable algorithms for network analysis and trying to understand microbial interactions in communities. And more importantly, in the context of today's talk, we study large networks and large multi-omic data sets and things like that. And we very closely work with the Center for Integrative Biology in Systems Medicine and also the Robert Bosch Center for Data Science and AI, which are two really up and coming centers in the institute. And these are some of our key research contributions. So we've contributed quite a few algorithms and theory in terms of understanding various kinds of networks, metabolic networks in particular, and so on. And also a bit on algorithms that focus on identifying important mutations and things like that. So all of these, I'm going to talk about few of these today, but you can read about more of these from my website. And the applications that typically excite us are in terms of metabolic engineering and also trying to obtain biological insights into various kinds of interactions and so on. Particularly, and now of late, we are very, very focused on microbial interactions in microbiomes and things like that. And all of this work is possible because we have a wide array of collaborations, both within the institute and outside. And these are some of our collaborators, and they played a significant role in developing the work that we do in our lab. The sampling of ongoing projects for those interested. So it just gives you a feel of what is it that we've been working on. We work on studying metabolic networks for metabolic engineering, trying to understand how metabolic networks function, what is their redundancy, how do they switch under different conditions. or trying to understand the infant gut microbiota through metabolic network modeling, and trying to understand the design principles of various kinds of networks and communities, and a few aspects of the theoretical synthetic biology. I'll not talk much more about it today. And but most importantly, we are very focused on trying to understand the community structure and microbiomes. Although today I'm going to talk about a slightly different topic based on some of the discussions I had with Professor Ashish. I'm going to talk about network biology and focusing more on machine learning or artificial intelligence to answer biological questions. Okay. And a quick word about our Center for Integrative Biology and Systems Medicine. We are an interdisciplinary group, and we focus on innovative approaches and algorithms to integrate multi-omic, multidimensional data to predict and manipulate complex biological systems. And we focus mainly on fundamental understanding of cells, but also ultimately looking at applications in personalized medicine. And we work very closely with the Robert Bosch Center for Data Science and AI at IIT Madras. So do check us out. Look us up on social media, follow us on Twitter and LinkedIn. These are some of our key research themes at the center, not very dissimilar to my own. We look at biological network analysis and then algorithms related for machine learning, graph mining, linear programming, and so on, applying it to study various interesting systems, some of which I'm going to talk to you today. Okay. And this brings us to today's talk. I'm going to give you a brief overview of whatever I can. I think I will go on until maybe 2:45. I want to give sufficient time for some discussion to follow. And so please have your questions ready. I would like to have quite a bit of discussion on the various concepts that are discussed today, and also any other generic questions or pointers you may have about the field in general, or application of AI or network science to biological problems and so on. Okay, with that, let me go to a very interesting problem, which is that of identifying disease modules in biological networks. Now, all of us are very familiar with networks. In fact, if you used Google Maps today, you've already used a graph theoretic algorithm on your commute to your from your place of living to your place of work or where you study and so on. And when you have a network that looks like this, the modules sort of declare themselves. It's very obvious, right? So you may have you may feel that these look like modules and so on, whereas here they look less obvious. And but the question is, how do you tease out these modules from any network to begin with, but more so biological networks? So graph theory or network science has many applications in biology, and there are many biological problems that naturally map onto graphs. For example, pathfinding in metabolic networks, or predicting protein-protein interactions. In fact, the same algorithms that are used in, say, Facebook or LinkedIn, especially in Facebook, where they suggest friends, right? So, do you know this person? The same algorithm is used in protein interaction networks to predict if two proteins will interact and so on. And then we also, and something as fundamental as read assembly of sequences from a next generation sequencing machine is not possible if without graph theoretic algorithms. Of course, you don't need it for assembling against a reference, but if you have like a new species that is being sequenced, or you want to rebuild your reference, you need to go in for graph theoretic algorithms. But today I'm going to talk to you about a few topics, starting with the identification of clusters or modules in large biological networks, which is also known as community detection. So how do you detect a community of people, or a community of nodes, or a community of related entities in a graph versus the other? Okay. And network biology, the term has gained widespread popularity. I would encourage all the students to read this beautiful review, which gives you a lot of information about all of these concepts. But today I'm going to focus on machine learning because it's become very a hot topic, and a lot of students want to know a little bit more about machine learning and how it can be applied to biology. Right? All of us know that your Amazon or Netflix is applying machine learning to sell you products and things like that. But how do we adapt machine learning algorithms? How do we leverage machine learning algorithms in the context of biology? There are various kinds of networks in biology. Okay, so this is sort of unraveling the social network underlying a cell. You have all these different molecules which interact with each other in very complex and interesting networks. You have metabolic networks, you have protein interaction networks, you have gene regulatory networks, signaling networks, and so on. Okay, I'll not talk about protein structure networks or the food web, but if you look at intracellular networks, there are already so many networks that exist within a single cell. And the cellular function itself is sort of a consequence of all of these very interesting underlying networks. Community detection in graphs is a very classic problem in graph theory. And you can see this network by the side. A very interesting backstory to that. So it's always nice to talk about it. In fact, there is also a sort of a prize for talking about this at a conference. There is something known as the Karate Club for somebody who's curious about these kinds of funny stories. Essentially, in 1977, there was a karate club that was existing in the US. And there's this Wayne Zachary, who was a social network scientist, who was trying to understand this club. So he figured out who was whose friend. There was no Facebook then, right? So this person number 25 was friends with 26, 28, and 32, and so on. Right? And you can see that this person 3 is a very gregarious person, is friends with as many as 8 or 9 people. So he was building this data set of who's friends with whom, and it turned out that there was a fight in the karate club, uh, between infighting between these people, and the club split into two. And it turns out that beautifully, that this was the split, right? And this could be very well inferred from the kind of connections that people had with each other, the friendships that people shared with one another, and so on. So this has today become a sort of gold standard, at least in computer science circles, for testing community detection algorithms and so on. But crudely speaking, you want to understand what are the nodes that are more tightly connected to one another than with the rest of the community, the rest of the network. Okay? And there are many algorithms that exist, but can you apply them directly to biological networks? Okay? So very crudely speaking, you can think of these as nodes that are tightly knit. So you can say that this is one tight group of nodes and this is one very tightly knit group of nodes. Why? Because they have more connections amongst themselves than to the outside world, so to speak. And this is, for example, a list of this is a bibliographic network, which essentially captures how papers cite one another and so on. So you see, statistical physics papers mostly cite one another, but they occasionally cite papers from some other fields and so on. So this is how this works, and this also helps to make networks into more tractable, smaller networks, and so on. And this is a wonderful book, but it's very heavy. It's for more for computer science people who are very interested in networks. But sure, anybody who's very passionate about network science can certainly read the first few chapters at least. Yeah. So why are we talking about modules in? We talked about community detection in a karate club. So there are some practical applications for that. But what about biological networks? It turns out that many functions in a cell are carried out in a highly modular fashion, and there are there are very functional units that work together to achieve various kinds of functions. Right? And many of these modules may so if you look at life prior to the genome sequencing project, people thought that once you sequence the genome, you will have a fantastic understanding of most diseases and so on. But it turns out that, well, it only answers part of your problem, and there are still many diseases that you cannot understand because they are not simply monogenic diseases or diseases that arise from the dysfunction of a single gene, but they arise from a breakdown in the network. The network is no longer functioning. So And as a consequence of that, there is a cascade of events which further disrupts the entire functioning of a particular cell, or then consequently a tissue or organ, and so on. There are many modules that are present in biological, various kinds of biological networks, and they typically show up as tightly connected nodes that are more connected than you would expect them to be connected just by chance. Right? So this is statistically speaking, because you always want to have a statistical significance associated with the observations that you want to, the conclusions that you make. So here you have nodes connected to each other. So if you were to randomly connect them versus they were connected in this fashion, are they significantly different? Is there more structure? Are certain nodes talking to each other more often than they would if they were just connecting randomly? Answering this question helps us find various kinds of groups of nodes in these networks and try to understand the basis of disease and so on. Okay. And modularity itself is a very interesting design principle in both engineering and biology. You see that there is always some level of modularity that's built into engineering systems, but and they're very much widely prevalent in biological systems. So how do you go about module detection in biology? And I'll talk about a particular challenge that we participated in. It's a very interesting problem. They gave us 6 networks which spanned various kinds of molecules. So one was a protein interaction network, 2 protein interaction networks, in fact, a signaling network, a co-expression network, a cancer dependency network, and ultimately just a simple homology network of which protein is similar to which other protein, and so on. Okay. And there were 2 challenges in this. This is an international competition, and they wanted us to identify modules in the individual networks at first, and also across all the networks. And they evaluated the performance by a complex process using genome-wide association study data sets and so on. I'll not get into those details. But the challenge was they gave us anonymized networks. And so you have no idea if you're picking the right modules or not. And so there's no way to sort of, if like in a supervised scenario in machine learning, you basically learn from examples. Somebody says that this kind of a person is likely to develop a particular kind of disease. And then you find a new person who has biochemical parameters or whatever genetic parameters similar to the person with disease. You may conclude that she or he may be likely to develop that complication. But here there is no ground truth available, which means that you have to try to look at some network structural properties and figure out that if these nodes are more tightly connected to each other, and are they going to work together, and so on. So we didn't know the biology of any of these nodes. We just got 1, 2, 3. I don't know if it is STAT or JAK or P53 or any other protein. So we got this kind of a network, and we had to say, okay, some of these nodes, some of these components of the network form a module, and so on. And we had to identify modules of a very specific size, from not less than 3 proteins and not more than 100 proteins, and so on. And we could also not find overlapping modules, meaning that you had to put every protein into only one module and not, but sometimes, in biology, that the same protein functions towards multiple diseases and so on. But here you have to sort of put them down in a single network. And the question we set about to answer was, how do you extend community detection algorithms to biological networks? And this is a wonderful challenge we participated in, where we had about 75 algorithms contributed by 400 participants across the world. And this was like a consortium paper that we were involved in. And more details of our own method are present in this paper. So it's a very interesting sort of read, even for a biologist, just to see how one goes about these kinds of problems, trying to break it down and infer interesting biological insights from structure and so on. Okay. And I was really fortunate to work with a couple of really bright students and some really fantastic collaborators that you see here. So Himanshu Srinivasan Parthasarathy from Ohio State, and also Professor Ramindran, who heads the Robert Bosch Center for Data Science at AI. So what did we do? We took three approaches. And we tried to first extend classic approaches and then identify core modules which are more structurally sort of closely knit. And then we also looked at what happens if you start identifying overlapping modules and so on. And we use some baseline algorithms. I'll not get into the details. I know this audience is more oriented towards the biology, but I'll just tell you that we applied some classic approaches and then identified certain core modules or compact modules in these communities which have greater relevance. Okay, and we looked at consensus across multiple algorithms. Well, that seems like a very reasonable thing to do. The way we did it was a very interesting approach. And one particular thing that would be well appreciated by a biologist is that we try to identify persistent communities based on perturbations. So essentially, what we did was we took this network, or let me just step through it in a while, and we also tried to break down large communities into more closely knit communities and so on. So first is, how do we do the ensemble clustering? So we have multiple algorithms. So we have different. Think of these as different proteins, and these are different algorithms, right? So one algorithm says that node 1 and node 3 are present in the same community, whereas node 2 is present in a different protein. 2 is present in a different. It is probably influencing a different disease or whatever. Whereas the second algorithm says that all three fellows are in the same community, and the third algorithm says that all three are in different communities. Okay, but then if you look at a single node, in N1 and N3 are in 2 out of 3 algorithms are said to be in the same community. Okay, so this information is used, and we then build a sort of similarity matrix, which is what is the probability of these two nodes being in the same community. And using this, we cluster them into communities and so on. I'll not go into further details. And we use an approach called modularity maximization that tries to find tightly knit communities and so on. Okay. This is the approach I was talking about, a very interesting approach. We said you take the original network and randomly drop a few edges, right? Like 1% of the edges, and do it 100 times. This will generate a large number of different networks, right? And each of them will have their own community structure. And now you use that community structure as an ensemble and again proceed in the same way as we did in the previous slide. And this gave us very interesting communities. And then we took a large community that was predicted by the standard algorithms. Standard algorithms they look for certain things in a community structure, and they give you large sort of groups of nodes that are in the same community. But in biology, you typically find that there are not more than say 50-60 genes that are dysregulated in the same disease and so on. So we tried to prune these modules to more tightly knit modules using certain approaches. For example, we try to identify how many outgoing edges are there for a particular vertex versus the number of internal edges and so on. Basically trying to understand what is the role of a particular node in the context of the larger community, and use that to fragment these communities. Okay, and this was the final result. And these are all various approaches that we had, and this was the winning approach, which was known as diffusion state distance. So we sort of beat the winning approach in at least 2 of the networks, and overall our approach had a much better hit ratio. So so you see, in all of these cases our hit ratio was superior to the winning. You don't see any bold entries in the last column. Okay, and so we were in the final scoring, we went a little bit below the the original the winning algorithm, but overall we made a very good we broke a lot of new ground in this area. Okay, so the application, of course, of this approach was to identify disease networks in terms of what are the nodes, what are the proteins that are dysregulated in a given disease and things like that. And very interesting that we could do this on a completely anonymized network with no biological information. This essentially means that this is a baseline, and you can do way better if you start including biological knowledge of or constraints to further inform your clustering. With that, I'm going to switch gears completely and talk to you about a very different problem, something that a lot of you are familiar from school days. You have a molecule on the left hand side, you have a molecule on the right hand side. How do you convert it? I'm sure in school this question was something like you have some phenol or like benzene on one side, and you have like 2-chlorophenol on the other side, and you have to figure out how to make this. And you have a very nice memory of organic chemistry reactions, so some of you may have dreaded doing it, but you would then say that, oh, I convert benzene to phenol using this particular reaction, and then I convert phenol to 2-chlorophenol, the substitution through another reaction, and so on. Okay, how do you do this whole kind of question now biochemically? Let's say I have asparagine and I want to convert it to something else or whatever, right? I want to create this is a carboxylic acid or whatever. How do I convert a given molecule A to a molecule B? Or maybe I know how do I produce this? Maybe this is a drug molecule, not this particular molecule, but suppose I have a drug molecule. I want to see if there is a biochemical way to produce this molecule. Can you predict a pathway for producing this molecule? Okay, I'm basically going to show you a very different application to essentially enthuse you towards applications of A, graph theory, and B, machine learning in solving biological problems. Okay. So today you have about 10,000 plus reactions in various biochemistry databases, which essentially capture what kind of conversions that known enzymes can catalyze. And by sort of copy pasting these non-native reactions into organisms has been a big cornerstone of metabolic engineering and driving the retrosynthesis of novel metabolites. This process is called retrosynthesis. When I say I have B, what do I produce it from? Right? Can you find the A from which I can produce my B? Okay, so this is because I have to retrosynthesize it, go backwards. And the question we set out to answer was, can we build a fully automated engine to predict pathways between two molecules, focusing or relying only on structural information? I do not know the pathway mechanisms. I do not know the delta G of formation. I have no other information. I know what molecules. I know that glucose plus ATP will give me glucose 6-phosphate plus ADP, and that is catalyzed by hexokinase enzyme. I know these kinds of things from biochemistry databases, but I really do not know if this is like some particular substitution reaction or what is the mechanism, what is the intermediate, and things like that. But I know the structures of all the components, all the compounds that are involved. So we built a very interesting approach called reaction miner, where we represent every molecule as a graph. So forget about protein networks for this part of the talk, and now your ethanol becomes a graph, and every molecule is represented as a graph, and we use this information to basically learn what is the kind of transformation that's happening in a reaction, right? Meaning, suppose you have a reaction A plus B going to C plus D, what is the pairing? Does A become C or D, right? So glucose becomes not ATP in the ADP in the hexokinase reaction, but glucose 6-phosphate. And ATP converts to ADP. This sounds very obvious, but how do you do it computationally when you have like thousands of reactions and so on? So how do you map them? And what, or if you have like a simple alcohol dehydrogenase, it takes ethanol and converts it to acetaldehyde. So how does this reaction happen? What does alcohol dehydrogenase look for? So we assume that it looks for a particular signature. It looks for like an alcohol group and some sort of chemistry around the group in terms of neighboring atoms and so on. And it finally, converts it to an aldehyde through a process which we do not know, which we don't worry about. We know the structure of these molecules, and we basically extract rules, right? So when I see these kinds of atoms, this bunch of atoms can be converted to this bunch of atoms by this kind of enzyme, right? So an alcohol is converted to an aldehyde through an alcohol dehydrogenase. So we put in rules like this, and the moment we put in rules like this and sort of embedded them into another network of rules, it was very simple for us. The nodes connect various reaction rules, and the edges connect rules that can be successively applied. So I find a molecule with a particular signature or product B of interest, a drug of interest or something. I can easily backtrace. So this must have been synthesized from one of these previous products using one of these enzymes. So I can literally just do a path trace on this network, and we came up with a very nice way to do that as well, which gave us very reliable results. And that is something that we could achieve, and we could get very good predictions of biologically reasonable pathways and so on. And this work was done by a very bright dual degree student, Aravind, and also and I did it in collaboration with Professor Shayan Ranu, who's now at IIT Delhi, but he was at IIT Madras when we did this work. And very interesting piece of work, using, because the 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 biological logic that we use is fairly simple, but the kind of data structures and algorithms that we needed to implement it was very fascinating. And it shows how biology has become very collaborative, and it requires a lot of interesting people with interesting different backgrounds to come together and work on challenging problems. But then I switched to another kind of problem solved on protein-protein networks. So this is, the network is similar to what I spoke in the first problem. But then the problem we solved was very, very interesting. How do you predict an essential gene in an interaction network? So which genes are essential for survival of an organism? And you might have seen this network picture, colorful network picture on my first slide as well. And this comes from a very classic paper in 2001 in Nature, where they laid out experimental data. So, so the yellows are basically those they didn't have data for, but the other colors that you see, like green, orange, and red, are essentially proteins that are essential, or slow down the growth of the organism and delete it, or non-essential, and so on. Okay, so what they showed was that there is this notion of centrality lethality, meaning nodes that are very centrally located in a network are more likely to be lethal, meaning they are like something like influential proteins. And in infrastructure networks, you can think of airport hubs and things like that. Right? A shutdown in an airport hub will cause severe repercussions in the entire network. And in fact, we had a very interesting paper on that recently, and it was on some of the press recently. So you can just look it up for the last couple of days, wherein we say how the spare capacity of networks can be exploited to prevent attacks and things like that. Now, cycling back to our problem at hand, can we predict if a particular node is essential or non-essential based on its neighborhood, its network neighborhood? And this is something I did again. You're familiar with these really smart people I work with, both Karthik and Professor Ravindran. And we used a bunch of network features, right? So looking at how many nodes a particular node is connected to, and in fact, things like page rank, which Google uses to say which page is more likely to be interesting to a user when they're searching for a particular term, and so on. So we use a lot of these so-called measures of centrality, and using labeled data from an experimentally determined source, we came up with several features that could, and we tested several features and finally came up with just a bunch of 12 features that can very accurately identify if a protein is likely to be essential or not. And this is very helpful for biologists, because they can base their experiments on this and build further hypotheses for experimental validation on this. And we essentially took about 2,700 interactomes from the string, and we put them through this pipeline, and we made predictions. You can see this on this website, and this is how the website looks. So it's a recent paper where we talk about the development of this website. So you have several organisms where you can just download it as a CSV and also look at what are the exact genes that are essential and so on. Okay, so this is another very interesting piece of work. So far I've given you uh, a quick overview of 3 different problems wherein we learn on networks or from networks or using networks to identify to solve various interesting biological problems. So I'm just going to switch gears slightly here and the moment. Not sure my PowerPoint is frozen. Just give me a second. So I'm just going to talk about.
Your PPT is not visible.
I know. My PowerPoint had an issue, so I'm just getting it back online. Give me a moment. I hope. I hope it's back online now. You're able to see my slides?
Yes, sir.
Yeah. Okay. So let me continue. So I'm going to now finally talk to you a little bit more about genomic data analysis, because I think that's something that many of you may be looking at careers in, and so on. And we have a very interesting approach to identify tumor suppressor genes and oncogenes using by studying functional mutations. Okay. And I don't need to tell this to this audience, but cancer is a disease of the genome, and you have various mutations or structural alterations, and lots of genomic studies that have shed a lot of insight into the development of cancer. And there's this notion of driver and passenger mutations in cancer based on what are the how they affect the proliferation of cancer cells and so on. So you have tumor suppressor genes and oncogenes. I'll not get into that, but typically what you find from most algorithms is that they try to identify frequently mutated genes and sort of capture them as important genes for cancer. But then there are lots of rare genes, especially some really rare genes, which are still very important in cancer, and you never end up finding those. Okay. And this is also captured in this figure. You may not be familiar with these terms, but precision essentially tells you what fraction of your predictions are correct, right? And in that way, they all perform well. But recall tells you what are the fraction of correct uh, things that you've actually captured, and that is very poor, meaning I have left whatever I predict is decent, is good, but then there are a lot of other genes that I have not predicted which have just completely missed, like too many false negatives. I have very few false positives, but too many false negatives. Okay. So how do we reliably predict cancer driver genes? And here I work with Malvika and who's my PhD student, and Professor Raghu, who is an expert in machine learning and data science. And we put all the data that we had through this elaborate pipeline, and we built a bunch of new features for these models and so on to predict what is going to be the impact of each and every mutation and things like that, and ultimately predicted several novel genes. Okay, and I will just cut to the chase. And what we did was, by building a careful uh, classification pipeline, we could predict several tumor suppressor genes and oncogenes. Some of them are known, and many of the others are very promising. And we have a bunch of genes of interest which are essentially very useful for biologists to start asking more questions about. And our method performs is able to capture genes which are mutated in very few samples. So they are not like commonly mutated genes, but are easy to capture as driver genes and so on. Unlike these genes which have to be which have very low mutation rates but are still very important in the development of cancer. And in another recent study, what we showed was very fascinating. We took similar data, and but what we asked was, you have a mutation like this, and by looking at this neighborhood of this mutation, what are the sequences on left hand side and right hand side, upstream and downstream? Can you still predict if there is a passenger or driver here? Okay, is it important for cancer and so on? And this was again fantastic work that was done by Shayantan, again with Professor Ravindra. Uh, and finally, I'm just going to give you a toolkit that we've recently developed. And for those of you who are looking to tinker around with genomic data, you can see this figure shows that there's a lot of complex steps involved. And if you want to get around all of this complexity, you can use our tool called iComic, and which is basically which can do all of this, right? It can do alignment, variant calling, and also run custom algorithms that we have developed for if it is cancer genomic data. And it's a simple point and click application that incorporates a dozen plus tools, and very user friendly, open source, and you can see very nice documentation on this website. I would encourage all of you to take a look. And I'll skip the tools, but I'll just show you how it looks. Right, so you can look at DNA sequencing pipeline, RNA sequencing pipeline, or even our CTAG and NB driver that I just described to you. And you can put an entire folder of files to run on, and use a bunch of threads if you're working on a parallel machine. And that's it. So everything is automated. So, and even the installation is like really simple. You can just do conda install iComic. So this really makes life easy for anyone who wants to just tinker with it without much programming experience and so on. Okay, this sort of brings me to the end of my talk, and just a few challenges, right? uh, First is that all these systems are on different scales. So you have some processes that are very fast, some are very slow, and so on. And you have length and time scales in biology. You have some large molecules, some big macromolecular chains, and some tiny signaling second messengers and so on. And the networks involve all kinds of molecules. So how do you get them together? How do you analyze them? How do you study them? And well, but thankfully, the importance of quantitative analysis of biological data is being recognized now, and for answering questions with respect to disease. And disease is a network malfunction problem. So you have to understand the network if you have to understand the disease. And this involves very close interaction between experimental biologists, engineers, data scientists, and so on. Okay. And there's just so much of data available, right? So you're just looking at, say, genomic data is not even the tip of the iceberg. You have transcriptomic, proteomic data, metabolomic data, and, well, not to mention all your microbiome data, gut metagenomics, and things like that. So a whole lot of data that's available. And I'll sort of close with this quote from Donald Knuth, who's one of the most famous computer scientists. And he says that he can't be as confident about computer science as he can be about biology, and how biology has 500 years of exciting problems to work on. And I think so. All of you are in a very exciting position to. You may not be fully interested in quantitative or computational biology, but you can surely start picking up some of the basics so that you can talk to other people who do this kind of work, and how they can help you in designing better experiments, coming with better hypotheses, or and so on. So with that, I'd like to acknowledge all my collaborators and wonderful students, and also our funding agencies, particularly the SERB, and which encourages us to talk to other universities and other colleges as part of our grants. So I'm very happy to acknowledge their support. Thank you all for listening, and do look up our webpage, and if some of you are interested, you're very welcome to join us, either for an intern or for a master's or a PhD. Thank you.
Okay. Thank you, Dr. Raman. Really, it's a very, very worthy and very insightful talk. Uh, Thank you for being with us. And basically, I'm a physicist, and I have one question. That's not a question. That's my, some kind of a dilemma in my mind. I hope that you will sort out this problem, which is going in my mind. Actually, I have a little bit of experience over the reaction mind during my PhD days, and I want to ask that if the model, or if the either the network modeling suggests us the one or two more pathways to solve out the exact reaction. So what should you take in this aspect? If your model is giving you one or more two pathways for for getting a best results.
Right. So we built a ranking scheme. So that the reactions would be more biologically plausible. They would show up higher, and we did this by using like a simple heuristic. We said that the pathway should try to, and we validated it by looking at known pathways, and we found that the top pathways were always ranked. The biologically plausible pathways were always ranked in our top few hits because of the heuristic that we used. It's essentially saying that every transformation that you do has to be small, but which probably takes care of the energetic issues and things like that, but should take you closer to the in the direction of the end product of interest and so on. So we basically mathematized that formulation, and using that we were able to get there are obviously multiple pathways we find, and some of them may not be relevant, and I usually include another quote in my talks, that all models are wrong, but some are useful. And the same way, there may be some mispredictions and so on, but overall they give you many ideas that you can finally test out in the lab. And this nothing better than computational approaches to tell you to plan your experiments on, right? Because they give you from a universe of possibilities, it reduces it to a smaller tractable number of possible experiments you want to try out in the lab, and come up with exciting biological results at the end of the day.
Yes, thank you. You have talked about the rank actually. Generally, in kind of a prediction model, we used to uh, some kind of a parameter, just like a standard error of estimations, and how many PLS vectors we are using to build up the models. So what kind of a parameters you are using in networking models?
Yeah, so here we basically used everything was related to the structure of these molecules, right? So we use the structure of these molecules to predict which pathways are going to be more likely to uh, or more important for more likely to work in a biological setting compared to other pathways and so on. So our rank was a measure of sort of biological plausibility
Okay, and
based
simultaneously
on
the
the
cross.
structures. Based on the structures, we didn't have any energetic information and so on. And if you do have something like delta G, that might be like a much more robust measure. But we showed that even in the absence of things like delta G, you could have fairly good performance and so on.
Yeah. And simultaneously, I think the cross validations should be going on simultaneously, and the validation in a laboratory scale, where the theoretical results match to the practical ones.
Absolutely, right? I think all of these, in fact, we work a lot on generating this hypotheses and then we work with experimental labs. So in this case, we basically compared against literature and so on. But in many of the other cases, especially in metabolic engineering and so on, we in fact, some of the questions are posed by the biologists saying, okay, how do I improve the yield of this particular molecule in this organism? So what are the kind of, how do we manipulate the network? And then we build some models and come up with predictions on these are the kind of reactions that you may want to overexpress in this network so that it improves the production of your molecule of interest. And we have validated some of these things in the lab as well. And this is a very central aspect of computational biology, wherein A, you generate hypotheses, and B, you experimentally validate them to and build more confidence in your models.
Yeah, really, the perfect model, it leads the less energy consumption in the reactions. So I have to ask you these questions to you. Somewhere, if we able to understand the perfect model for the perfect reactions, definitely
Yeah.
we will move
Yeah.
towards the less energy
Exactly,
consumption.
right. So what does biology really optimize? What does nature
Yeah,
optimize? And this is a very interesting question. So does it optimize for delta G? Does it optimize for length of the pathway? Or does it optimize for ATP production? So we've been asking this question, and so for instance, something like glycolysis seems to be the shortest pathway that produces a reasonable amount of ATP. So there are shorter glycolyses that are possible. So there is in fact I think a three-step glycolysis that was found in some organism or was engineered in another organism. But if you look at amount of ATP production and so on, that's what makes it interesting. So there are multiple constraints and of course evolutionary accidents and so on. So there are multiple constraints that cells seem to be optimizing, and it's very interesting. We still don't fully know these constraints, I would say.
yeah. Thank you, Dr. Aman. And one last query from my mind. That is you know, uh, you are in the position where you connect the computational approach to the biological things. Okay, but when the students are generally in the undergraduate and post graduation, that time they separate to both things. Computer is on one hand, as well as biological is on one hand. How we can motivate them to go interfering the computational approach in a biological system?
Yeah, it's a very good question, and I think these I teach a course on systems biology, and I have students from different backgrounds who take that course, and everybody has to do a project in that course, and they have to do it in teams. And I encourage them to team up in such a way that one of them has has a stronger computing or programming skills, and the other one has a stronger biological skills, and so on. And this sort of collaboration helps them to understand each other's perspective and get more hands-on experience and so on. So the short answer to your question is that this happens by doing short internships or short projects by actually experimenting. It is not difficult. One might be have a mental block against say trying to do computation or something like that. But when you start trying it out, especially with people with classmates and from another department or another place who are similar age and similar thinking and so on, but have different skills, you can really quickly learn from their complementary skills and so on. And I also teach an NPTEL course on systems biology,
Yeah,
and I would really encourage people who are interested to take a look at least at the first few weeks where we talk about networks and so on. And it will show you that yeah, networks are sort of complex, but there are easy ways to break down the concepts and understand them.
yeah. Most of the students, a lot of the students enrolled that particular course from our college also, which you
Good
have
to know.
instructed. That's a wonderful, So once again, thank you, Dr. Aman. Now I would like to invite Dr. Dipti Gulati for vote of thanks. Please, ma'am. Now mic is over to you.
Thank you, Ashish sir. A very good afternoon, everyone. It's my privilege to have been asked to propose a vote of thanks on this occasion. I, Deepthi Gulati from the Department of Biotechnology, On behalf of organizing committee, the students, and on my own behalf, extend a very hearty vote of thanks to Dr. Karthik Raman for presenting in the most interesting and meaningful way the design principle of biological systems through computational modeling. I'm sure,
Hello.
sir, your talk must have ignited interest of students in the field of system biology. I would also like to congratulate Dr. Ashish Raturi and his entire team for organizing this wonderful talk. I would like to express our profound gratitude to our chairman, Sri Arvind Gupta ji, for his constant motivation to conduct such events. I would also like to thank our principal, Dr. Shelja Pant ma'am, for her support and guidance.
Hello.
I'm very happy to express the vote of thanks to our IT support team for the technical arrangements. Last but not the least, I thank all the wonderful students who have turned up in such great number for this program. Once again, I thank all for your cordial cooperation. Thank you, Ashish sir, for
Dr.
giving me
Ashish,
the
I'm not able to hear
opportunity
anything.
to deliver
I
this vote
think.
of thanks. Thank you.
Yes, thank you, ma'am. Yes, Dr. Karthik, are you going to speak something?
No, no, no. I couldn't hear anything, so I was just wondering.
Okay, I think maybe some internet links over the
Okay, okay. No worries.
But now I would like to thanks again Dr. Karthik Raman and to all audience to being with us. And one request for to Dr. Raman that whenever you visit Uttarakhand, please visit us at our college. We are located in the foothills of the Himalaya and surrounded by the lush green forests. So
I
we would
can
like
imagine.
to thank you,
Sure.
sir.
Thank you. Thank you so much.
Thank you. So we are signing off until we meet again. Thank you very much.
Yes, sir. Thank you. Bye bye.